Welcome to today's webinar, Effective and Efficient Approaches to Cell Line Engineering in CHO Cells. I'm Nico Cavello, Product Marketing Associate of Next Generation Sequencing Services at GeneWiz, and I'm very pleased to introduce today's presenter, Stephanie Sandifer. Stephanie received a BS degree in microbiology from Purdue University and a MS degree in microbiology and immunology from Vanderbilt University. She joined Eli Lilly and Company in 2000, beginning her career in the Discovery Organization, specifically the Department of Infectious Diseases, developing a mouse model in which to evaluate anti-HCV compounds. Stephanie later transitioned to bioprocess research and development in 2003 to develop microbial expression technologies for the production of therapeutic proteins. In 2007, she moved to the Molecular and Cellular Biology Group, which is responsible for supporting all of Lilly's mammalian-based manufacturing processes. Most recently, Stephanie has helped develop and establish state-of-the-art cell line engineering capabilities at Lilly. If you have any questions for Stephanie during the webinar, please enter them into the Q&A section, and we will address as many questions as possible at the end of the session. And now, please welcome Stephanie Sandifer.
I would like to thank GeneWiz for the opportunity to present to you today on effective and efficient approaches to cell line engineering and CHO cells. The production cell line is the foundation of bioprocess development. The quality of the cell line will impact key attributes such as productivity and product quality. Due to significant costs associated with cell line switches, there is a strong business driver to develop the commercial production cell line to support Phase 1 activities. Anything that can improve the quality of the production cell line will be advantageous in the clinical development of a molecule. Cell line engineering capabilities can enable commercial cell line development by improving certain product-related characteristics. However, cell line engineering can negatively impact development timelines in a time when every day counts. So what opportunities exist to minimize the impact of cell line engineering to a project's timeline? That is what I will address in my presentation today. There are several business drivers for cell line engineering. An array of genes can be targeted for disruption and can be grouped into three main buckets: product-specific challenges, general product quality challenges, and process-related challenges. To date, we have designed reagents for targets that fall into each of these main areas. There are several technologies available for cell line engineering. Based on having the clearest IP landscape, Lilly has focused on using the zinc finger nuclease or ZFN technology. However, we are very interested in investigating the RNA-based systems such as CRISPR-Cas9 and CFP1 due to the ease and quickness of generating the reagents. Before I get into my talk, I would like to introduce you to a few key concepts. The first is the concept of monoallelic versus biallelic mutations. Remember, in a diploid cell, and we're assuming that a CHO cell is diploid, there are two copies of each gene known as alleles. When ZFN-mediated double-strand breaks results in the formation of indels in one of the chromosomes, I will refer to that as a monoallelic knockout or mutation. If indels happen in both chromosomes, that is referred to as a biallelic knockout or mutation. If the mutations are the same in both alleles, I will refer to that as a homozygous biallelic mutation, and if the mutations are different in the two alleles, that is referred to as a heterozygous biallelic mutation. The next thing that I would like to introduce you to is the Surveyor Mutation Detection Assay, or MDA. The MDA is a PCR-based assay where a fragment around the ZFN binding region is amplified. The PCR product is then denatured and allowed to reanneal. If heterologous sequences anneal, that will create a bubble of mismatch, which is then recognized by the cell 1 enzyme provided in the kit. The cell 1 digestion products are resolved by gel electrophoresis. Those samples that show digestion products are said to be MDA positive and will contain one or more mutations. Once the MDA positive cell lines are identified, the next step is to characterize a subgroup by sequencing of the ZFN PCR product. Sequencing will provide information on the nature of the indel, including if the cell line is monoallelic or homozygous or heterozygous biallelic. The cell line engineering process flow is simple yet can be quite time-consuming. The first step is to design the reagent. Regardless of the technology used, we want to target the exons rather than introns or splice regions. It is preferable to target the 5' end of the gene. However, we do have reagents that target the 3' end of the gene that have been successful in disrupting that gene. For ZFNs, it can take 1 to 4 months to design and receive the reagent, although the average is closer to 5 weeks. One major advantage of the RNA-based technologies is the timeliness of receiving the reagent, where it takes about 3 weeks to receive CRISPR-Cas9 plasmids ready for transfection. Once the reagent is in-house, we are ready for transfection. We transfect using the ZFN mRNA rather than the ZFN plasmids to eliminate any possibility of integration into the genome. And like most things in science, we learned that one the hard way. Since we are transfecting mRNA, we've moved to using the nucleofector device and associated nucleofection kit from Lonza for transfection, as this seems to give us a higher transfection efficiency. Once the cells recover from transfection, the ZFN activity is measured by evaluating the bulk pool in the MDA. Only a bulk pool that shows a positive response in the MDA is processed forward to single-cell sorting. We use FACS technology to single-cell sort the MDA positive bulk pool into 384 well plates. Once cells grow out in the single wells, they are consolidated into deep well plates so that the ZFN PCR MDA screening can begin. For the MDA screen, it is important to ensure that the PCR product is clean, as extra bands or primer dimers makes it hard to interpret the results. It is also important that the PCR reagent is compatible with the cell 1 enzyme. 2 MDA reactions are performed, minus and plus wild-type PCR product mixed in order to screen for putative homozygous biallelic mutations, and I will address this in the next slide. Implementation of this kind of MDA screen can knock off 4 to 6 weeks from the cell line engineering timeline. Once MDA positive cell lines are identified, they are characterized by sequencing of the ZFN PCR product. The MDA alone cannot identify cell lines that contain homozygous biallelic mutations because the mutation is the same in both alleles. In the MDA alone, there will be only self-hybridization, creating a homoduplex, no mismatch, and therefore only one fragment will be seen on the gel. However, if a second MDA reaction is performed on the same samples, this time mixing in some wild-type PCR product, that will result in some level of cross-hybridization, heteroduplex formation, mismatches, and multiple fragments on the gel. It is worthy of noting that the MDA is not capable of distinguishing between heterozygous biallelic and monoallelic mutations. In either situation, MDA or MDA plus wild-type mix-in, there will be some level of cross or self-hybridization, heteroduplex formation, mismatches, and multiple fragments on the gel. If we can develop an assay that can distinguish between these two possibilities earlier in the process, we can shave time off the entire cell line engineering process, leading to less of an impact on a project's timeline. I'm currently working on developing and implementing such an assay, which would actually eliminate the need altogether to do the MDA. This slide shows an example of a 96 well plate undergoing the MDA. The left side of the gel shows the MDA, and the right shows the MDA plus wild-type mix-in. Now if we focus on the left side for a moment, we can see that there are several cell lines that show a positive response in the MDA. These cell lines contain monoallelic or heterozygous biallelic mutations. Remember, the MDA cannot distinguish between these two possibilities. Now if we compare the MDA result to that of the MDA mix-in, we will find that there are several cell lines, such as the one highlighted in the red box, that were negative on the left side, the MDA screen, but positive on the right, with the wild-type mix-in. This identifies this cell line as one that contains a putative homozygous biallelic mutation. If putative homozygous biallelic cell lines are identified, those can be characterized by directly sequencing the ZFN PCR product using Sanger sequencing because there will be clean sequence. If putative homozygous biallelic cell lines are not identified, then a subset of the MDA positive cell lines are consolidated to a deep well plate to prepare for characterization using next generation sequencing or NGS. Introduction of NGS has vastly improved the efficiency of the screening process. NGS is capable of resolving the sequence of mixed PCR products such as seen in monoallelic or heterozygous biallelic mutations. NGS is extremely high throughput and I typically screen 96 or 192 cell lines per project. In fact, for one project, I was able to screen 1,600 cell lines by NGS. With input on how I evaluate the NGS data, GENEWIZ developed the Genotyper Next tool that allows for quick evaluation of each cell line. Now, instead of sifting through Excel spreadsheets with 96 tabs containing the sequences found in each well, I get a pictorial representation of the plate. Based on the reference sequence provided, the level of wild type and mutant sequence found in each well is shown, and in fact the wells are color-coded based on this data. The wells colored blue are those cell lines that contain biallelic mutations, or in other words, no wild type sequence. The yellow wells represent monoallelic mutations, and the brown ones are wild type. For our purposes, we are interested in only the blue wells. You will note that there are two numbers within each well. The top number represents the percent mutant sequence, and the bottom number, a relatively new addition to the tool, represents the percent of the mutant sequence that contains out-of-frame mutations. So any well that contains less than 98% frameshift mutation are discarded from further evaluation. In a very short amount of time, the data has gone from needing to evaluate 96 wells to needing to further evaluate 9. Now when any of the wells is clicked on, for example C9, the data behind the well comes up to the right. There are several pieces of data shown, but I've highlighted the two most important ones. The endo-length histogram, which shows the nature of the mutation and the number of mutations present. In this case, there is a 7 base pair deletion in one allele and an 8 base pair deletion in the other allele. If there was an insertion, it would be shown on this side. Also shown is the alignment of the DNA sequence, which shows where in the reference sequence the indel happened, and in the case of insertions, what the inserted bases are. A separate Excel file contains the sequence in a copy-paste format and also shows the resulting consequence of the mutation as it relates to the protein sequence, only in the context of the reference sequence. So where in the clinical cell line generation process does host cell engineering fit? Shown here is a pictorial representation of the cell line generation process. From transfection to identification of the protection cell line takes approximately 27 weeks or about 7 months. The goal of this work is to fit cell line engineering into the process with the least amount of impact to the timeline. There are several steps within the cell line generation process where cell line engineering can be implemented depending on what one wants to accomplish or the specific application. For example, if the gene to be targeted is one found in the process-related challenge bucket, such as a host cell protein that co-purifies with many different molecules, the best track would be to engineer a new parental cell line, which would then be able to be used for all future molecules. Generation of a new parental cell line, minus any downstream evaluation, will take 2 months, assuming we have the reagent in-house, and this should be done before the molecule comes to development for clinical cell line generation. If, however, there is a product-related challenge, such as a host cell protein binding to the active site of the antibody, engineering can be initiated within the cell line generation process. Cell line engineering can be started after generation of the selected bulk pool or after completing the deep well shaken expression study. Engineering on the selected bulk culture will add 1 to 2 weeks to account for the timeline. It would add 1 to 2 weeks to the timeline to account for the ZFN transfection. However, the screen size at the deep well stage will need to be increased. This is because we are now looking for 2 events in the same cell. We need to find a cell line with high productivity that contains biallelic mutations. Alternatively, the cell line engineering work can begin on cell lines that show high productivity in the small scale screen. However, this will add 2 months to the timeline as we will need to go back to the single cell sorting stage and continue forward from there. Lastly, if a target is identified later, perhaps a general product quality related challenge such as proteolytic degradation, engineering can be done on the production cell line. This is the least desirable step in the process to introduce engineering because it will typically result in a cell line switch and everything associated with that. However, the issue could be severe enough to hinder developability of a molecule, and cell line engineering could be the only thing that enables commercialization. In summary, keys to efficient cell line engineering include early target identification and access to genome editing reagents. To date, cell line engineering and cell line generation needs to be done sequentially to ensure finding a highly productive cell line that contains biallelic mutations. However, work is being done to improve the screening process in the hopes for allowing simultaneous engineering and cell line generation. In addition, if there are multiple targets for the same cell, the engineering of each target should be done sequentially to ensure finding biallelic mutations in both genes. In this case, the MDA screening is a labor-intensive bottleneck, and as new screening processes are developed, it will be feasible to engineer multiple targets simultaneously, thus minimizing the timeline. Currently, it takes 2 months to target a single gene, and this represents the time from transfection to banking of cell lines with biallelic mutations. Any time associated with evaluating the cell line is out of scope of this timeline. The 2-month timeline assumes that homozygous biallelic cell lines are identified. We are actively pursuing targeted and/or semi-targeted integration systems for cell line generation. Implementation of these new systems will further reduce the impact of engineering to the timeline, in that it will now be possible to do cell line engineering and cell line generation simultaneously without increasing the screen size. By reducing the time it takes to engineer a cell line and being able to incorporate it seamlessly into the cell line generation process, it is feasible that cell line engineering, if required, can enable commercial cell generation for phase 1 clinical evaluation. I would like to thank members of our molecular and cell biology group, as well as members of the analytical group. I would also like to thank the management team, past and present, for their support of cell line engineering, and we would like to acknowledge Lonza for licensing and permission to modify the CHO expression system. Thank you for your time.
Thank you, Stephanie, for such an informative webinar on the effective and efficient approaches to cell line engineering. We had some really good questions come in throughout the webinar. The probability of obtaining a desired modification, and how large are these typical screens?
Thank you for the question. My typical screen size is 960 wells or 10 deep well plates, although I do have examples of screening more or less depending on the target. I've screened as few as 480 clonally-derived cell lines and as many as 1,600 clonally-derived cell lines. And that really depends on the target and specific application. The probability of finding an acceptable biallelic mutation is also target dependent. It's hard to give an exact probability because screening methods have evolved over time. Also, I typically do not screen all MDA positive cell lines, only a subset. The subset tends to be skewed towards those most likely to have a chance of being a biallelic mutation, such as screening mostly those identified as homozygous biallelic. It would be easier to give you a range of the MDA positive cells, which is also a target or ZSN specific. Depending on the ZSN that I've used, I've gotten between 9.1 to over 70% positive CDCLs in the MDA.
Thank you, Stephanie, for those great insights. We actually have another question coming in from Boston. How many independent targets have you disrupted? And have you ever been unable to generate a biallelic disruption?
Thank you for that question. So today we have disrupted 9 different targets, and we currently have reagents for another 8 targets in-house. Some of those have been single targets, and some have been targets that are planned to be multiplexed for different targets in the same host cell. The most I've ever gone after in a single cell is 5 targets, although there's one more target for that cell line that I would like to go after, which would be a total of 6 in one cell. Going after 2 targets simultaneously, I have to screen for both at the same time, and I have been unsuccessful at finding cell lines with acceptable biallelic mutations in both targets. In one instance, all I got out of the screen was monoallelic for both targets. In a second example, I was able to find biallelic mutations in one target or the other, but not in both. In the 5-target cell line, I was unable to find a biallelic mutation in the final target. That was one of the lower percentage ZSN reagents that I used at about 30%. And from that, 90% of those screened were monoallelic cell lines. The nature of the indels for that one leads me to believe that this particular target is lethal. However, it's not known if it's lethal by itself or in combination with the other disrupted targets. Other than that one example, I've been able to get acceptable biallelic mutations on all targets.
Thank you, Stephanie. Our next question is actually coming in from Alaska. Do you need to demonstrate stability of your engineered cell lines? If so, what does that process look like?
Yes, it is important to demonstrate stability of the engineered cell lines. This is mainly done by aging the cell lines through a typical manufacturing window time frame, which is generally taking them out to generation 60. The targeted region is then sequenced in an early, mid, and late generation to make sure that the indel is not changing over time. So far, I have found no indication of instability of the genetic modification for all the engineered cell lines, with the only exception being the one cell line that was engineered by using the plasmids for the ZFN rather than the mRNA. In that particular cell line, the ZFN plasmid was found to be integrated into the cell's genome, and over time, the ZFN was still active, creating slight modifications. For this reason, we only use the ZFN mRNA when transfecting.
Thank you, Stephanie. We have time for one additional question today. This question is coming in from Germany. Are there risks associated with implementing engineered cell lines, specifically as platform parental cell lines?
Anytime we move away from a platform process, which includes changing parental cell lines, there are potential risks involved. In the case for parental cell lines, those risks could include differences in the cell size or cell growth, transfection efficiency, or downstream applications, with potential differences in product quality or alterations in the primary recovery or purification. So anytime we're changing anything away from the platform, we need to do some studies to make sure that all of our downstream platform processes still work.
Thank you, Stephanie. So this is all the time we have today. I would like to thank Stephanie Sandifer for presenting as part of our GENEWIZ Week webinar series. Don't forget, along with our webinar series, we have other ongoing activities such as the Lab Startup Program, the GENEWIZ Credit Awards, and exclusive promotions. Make sure you check them out at genewizweek.com.